Welcome. Managing genotoxic impurities is a challenging task for pharmaceutical industries. That is why I decided to share my skill-based knowledge on this subject through this channel. I have prepared lectures on this subject in a sequential and progressive manner, and I am going to cover the same in a multiple sessions. This is the first lecture of this series, and in this lecture, I am going to discuss basic chemistry behind genotoxicity. In this session, you will learn what is genotoxicity, what is difference between genotoxicity and mutagenicity, which type of chemicals are genotoxic in nature, why epoxide, aromatic amines, and aromatic nitro compounds are genotoxic in nature, what is the structural approach to predict genotoxic nature of a molecule, will all molecules with structurally alert features be genotoxic in nature, what is the effect of substitution on genotoxicity nature of a molecule, what are the advantages of structure alert concept? Genotoxicity. Genotoxic, the meaning is damaging to genetic material. DNA can be damaged by various sources, and one of them is genotoxic or mutagenic chemicals. We are going to discuss in details. Now, question is, what is genotoxicity and what is mutagenicity? The key difference between genotoxicity and mutagenicity is that genotoxicity is the ability of a substance to cause toxicity on DNA or genetic material of a cell, while mutagenicity is the ability of an agent to cause mutations. All mutagens are genotoxic. However, not all genotoxic substances are mutagenic. Genotoxins can induce genetic mutation, chromosomal break, chromosomal rearrangement, cancer, or adverse effect at very low level. Types of DNA damage. DNA is a double helix formed by base pairs attached to a sugar phosphate backbone. Base pairs, sugar phosphate backbone. DNA may be damaged by cross-linking, oxidative, adapt formation, strand breaks. Any chemicals which can alter the identity of these base pair means adenine, thymine, guanine or cytosine can damage the DNA. Now question is, which type of chemicals can alter the identity of this base pair? To understand the same, we are going to discuss structure of this base pair in next slide. These are structures of thymine, adenine, guanine, cytosines. Means these are structures of bases of DNA. From structure, it is clear that each having multiple nitrogen atoms. Each nitrogen atoms contains a lone pair of electrons. These lone pair of electrons make these bases as a nucleophile, and hence an electrophile, that is any cation or molecule with cationic nature, may attack on these bases and can affect the identity of DNA. This means genotoxins are either electrophiles per se or be activated to electrophilic reactive intermediates in vivo. Hence, electrophiles or electrophilic reactive intermediates can make a bond with nucleophilic part of DNA and can damage DNA. Structures or functional groups of above characteristic are genotoxic in nature, and this is also called structural alert concept or SAS concept. A structural alert concept has been found one of the most useful scheme to assess carcinogenic potential of untested molecules. Hence, DNA may potentially interact or make a covalent bond with epoxide, aromatic amines, aryl nitro compounds, primary halide, alkyl sulfonates, ester, alkyl ester, aromatic aldehydes. Molecules of above class come under primary genotoxic impurities or PGIs. We are going to discuss in details in next slide. Now question is why epoxides are genotoxic in nature. The stained ring system of epoxides facilitate the generation of carbonium ion, and that carbonium ion act as a electrophile by the opening of the ring. That carbonium ion mediate with nucleophilic sites of the DNA and can damage the identity of DNA. That is the reason epoxides are genotoxic in nature. Now question is why aromatic amines or aromatic nitro compounds are genotoxic in nature. The aromatic nitro compound is reduced to aromatic amino compound in the intestine by metabolic activation. Aromatic amino compound oxidized to N-hydroxyarylamine, which is an electrophile in the liver. N-hydroxyarylamine react directly with DNA or be further activated through the formation of acetate or sulfate esters. This lead to blocked replication of DNA and induce mutation. That is the reason aromatic amines and aromatic nitro compounds may act as a genotoxic chemical. This is the pathway of the reaction mechanism. Nitro group is converted into NaOH or NH2 group, and these NH2 group or NaOH group having lone pair of electron, but this lone pair of electron is involved in the resonance and make NH2 group positively charged, which act as a electrophile and make a bond with nucleophilic part of the DNA and damage the DNA. That is the reason amine and nitro compounds are genotoxic in nature. Primary genotoxic impurity concerns. Aromatics. These aromatics compounds are genotoxic in nature. Alkyl and aryl groups. These heteroaromatic groups come under potential genotoxic impurities (PGIs). Now question is what is the effect of substitution on genotoxicity of a molecule? Each positional isomer is equally genotoxic in nature. Ortho substitution with functional groups of positive inductive effect can enhance genotoxicity. Ortho and meta substitution with bulky groups can suppress genotoxicity of a molecule. Planar molecules with electrophilic groups intercalate DNA better. Bulky groups ortho to an aromatic amino substituted amino or nitro group abolishes toxicity. Based on discussion, now you can predict genotoxic nature of these molecules. But keep in mind, this will be only prediction. Final decision will be given only after proper lab testing. For example, this aromatic amino compound contains two methyl group which are ortho to NH2 group, As we discussed, these methyl group due to plus inductive effect can enhance genotoxicity of this aromatic amino molecule. Similarly, other molecules can also be explained. This is all about basic chemistry behind genotoxicity. Any question related to this lecture, you can write in the comment section and I will answer on priority basis. If you are new on this channel, please subscribe to the channel and also give a like to this video. Thank you for watching this lecture.